Good afternoon, everyone. This is Clint Nesbitt. I am the Senior Director of Science and Regulatory Affairs with the Food and Ag section of the Biotechnology Industry Organization, also known as BIO. I've got 12 o'clock here on my watch, so I think we'll go ahead and get our seminar started. If you're not familiar with BIO, BIO is a Washington, D.C. based trade organization that represents many of the industry leaders working in food and agriculture. And as such, we are a very strong supporter of public investment in food and agricultural research. As such, BIO is a member of the National Coalition of Food and Agricultural Research, the sponsor of the seminar today. I personally serve on the NCFAR board and am pleased to act as the moderator for today's webinar. So this is now the fifth time in over 150 Lunch and Learn seminars where we are welcoming you to a virtual Lunch and Learn seminar via webinar. I think as we're all dealing with COVID-19 and social distancing and the like, this has been our approach to continue serving as a resource to folks on the Hill, agency officials, other interested stakeholders, and the like, until we are able to gather again in person on the Hill to share both knowledge and food. So in terms of logistics for today's webinar, we're going to keep all of you with your microphones closed, and we will have an opportunity for questions at the end. I would ask that you use the questions function within GoTo. That's in the little options menu on your screen. Type your questions in, and then I will read the questions to our speaker and moderate those at the end of the presentation. I would also just mention that certainly one of the commitments for this program is that we want to make sure that we end on time out of respect for your time. So I've asked our speaker to make sure he finishes his presentation by around 12:40 or so that we have time for Q&A at the end. The webinar is being recorded, so we'll circulate a link to this to you at the end if you're interested. And of course, the presentation itself will be published on the NCFAR website, ncfar.org. That's me. So just to give you a little bit of background about NCFAR, NCFAR is a consensus-based and customer-led coalition that serves as a forum and a unified voice in support of sustaining and increasing public investment at the national level in food and agricultural research, extension, and education. So our seminar series is intended to really feature those sort of leading edge researchers discussing their work and in meeting these 21st century challenges and opportunities and providing timely information that may prove useful in your own work. This is just a snapshot of the sponsors of the seminar series to give you an idea of the breadth of organization that are contributing to this effort. And now it actually gives me great pleasure to turn things over to our featured speaker of the afternoon, Dr. Rodolphe Barrangou. He is a professor of probiotics at North Carolina State University. You can see his full biography here on the screen, and we also included more details about his background in the invitation to the webinar. The title of Dr. Barrangou's presentation this afternoon is Catching Up with the CRISPR Craze, Applications of Genome Editing in Ag and Beyond. So please welcome Dr. Barrangou for this afternoon's talk.
All right, good afternoon, everyone. Thank you for having me. It's my great pleasure to be here, and I'm quite thankful for the opportunity to share some insights into some exciting things happening with regards to CRISPR. We are really witnessing in real time a technological revolution with the CRISPR craze. And I'm going to talk about the technology in general today, but specifically highlight the many applications of CRISPR-based genome editing in food and ag and beyond. So to give you an idea how far and how fast and how broadly this technology has led and empowered, arguably, the genome editing revolution, you just need to look at a small few numbers and a couple of maps to get a feel for how broadly disseminated and how quickly this technology has been disseminated across the planet. So as of right now, there's almost 200,000 labs across over 100 different nations that have been the recipients of CRISPR-based constructs distributed by AgGene, a not-for-profit organization in Cambridge, Massachusetts. And you can see that the countries and the geographies in which CRISPR research is being pioneered is extended to over 100 different countries, whereby those almost 200,000 labs, likely staffed with about 1 million people, 1 million geneticists, are very rapidly, very quickly, very powerfully using this technology in all kinds of cells to understand how organisms work and how they can have their genome edited or their genomes altered. To give you a little bit more granularity with regards to where we are right now, this is a very explosive field. So this is the number of papers and scientific studies that have been published on genome editing, not just CRISPR, but genome editing in general, in the last 2 decades. And you can see—
Dr. Barrangou, I apologize, I'm going to interrupt. We are not actually able to see your slides at the moment. Ah, there you go.
Do you see them now?
Yeah, I think if you project that now as a slideshow.
Do you see this?
I do not see that now. I can only see it when you're running in windowed view. I can see that.
Huh. All right, so you can see slides right now.
I can see the slides now, yeah. So I would propose you continue to run in that windowed mode rather than full screen. Sorry to interrupt.
We'll do it that way. It's all good. Thank you for doing that. So you see that right now? You see this?
No, I cannot see that.
So we'll just stick with that. That's good. So as I was saying, there's pretty much 200,000 laboratories staffed by around 1 million scientists in over 100 different countries that have access to this very powerful technology. And they've published extremely fast. So we're talking about tens of thousands of scientific studies that have come out in the last couple of years, showing and illustrating how those technologies can be used in all kinds of fields, including food and ag. So it's very quickly being diversified across different topics and themes for science. But what's important to note today is that there's over 100 different countries really leveraging, exploiting, and using this technology. The top 10 accounts for the large majority thereof. And conveniently, the US is still the #1 in the world. But at the current pace, you can start seeing that the pace of growth and expansion in China at current pace will probably overtake the US for CRISPR research in terms of number of papers by 2022. Another interesting point here, looking at geographical diversity, is the many continents that are represented in the top 10, with Europe heavily represented, Asia heavily represented with China and Japan, and then also Oceania with Australia being a top 10 country in the world looking at CRISPR research. This actually means that besides the science and the fundamental academic research itself, this is a competitive field also for business, the bioeconomy and industry and partners who are using those technologies and translating them into actual products. And again, you can see that the geopolitical interests of those countries in food and ag are certainly noteworthy across the board. So what I intend to do today in the next 30-ish minutes is to walk you through the CRISPR Craze. Where are we today, which you get a feel for right now? Where are we right now in food and ag in particular? And where are we headed next? What are the consequences scientifically, technologically, but also in terms of society, government, and industry? And what I'm going to walk you through is how the CRISPR science has turned very quickly into genome editing technology and how it's been applied into products that are being accepted and launched, hopefully, to the public. So scientifically, I'm not going to go into the details of CRISPR-Cas systems, their genetics, the genome editing mechanisms involved, but we all know and realize here in this room today or at home today at the office that the ag industry is being revolutionized. The future of ag is already unfolding before our eyes in real time with precision ag, with omics, bioinformatics, genomics, and the like, big data, machine learning, artificial intelligence, microbiome sensors, drones, and then certainly last but not least, genome editing as a very powerful technology that enables those hundreds of thousands of labs to actually breed next generation crops, next generation livestock, generate next generation bacteria used throughout the food supply chain. And we're talking about transformational new technologies that enable better and easier and faster and cheaper and more democratized and more accessible genome editing for all those scientists that have access to that technology. So let me just take a couple of minutes here to explain what genome editing actually is. You're all familiar with word processing. This is why it's called genome editing. You can actually edit or alter the sequence of DNA very precisely using a two-step process. Much like you would put a cursor into a word text, you can use CRISPR-based nucleases to very specifically locate and generate a cut, a cleavage event, at a sequence of interest in the genome of virtually any organism you can think of. So CRISPR is a cursor that enables people to cut text in DNA precisely, efficiently, predictably, and programmably. And then in the second step, much like you would edit text after you place a cursor, you can change the DNA sequence or the text precisely at the site of cleavage. This is why it's called editing. You can edit a letter from a G to an A precisely and predictably, or you can add a word or remove a word, or add a sentence and remove a sentence, or even change the punctuation in terms of transcription. So we now have a technology, CRISPR-based technologies, that allow us to essentially rewrite the genome however we see fit, conveniently, affordably, flexibly, and multiplexibly. And in the last 8 years, since the advent of CRISPR-based technologies and the first proof of concept that they can be used for genome editing in a variety of organisms across the tree of life, the genome editing toolbox has been improved and diversified and sharpened in so many ways, whereby the average geneticist in the average lab can perform genome editing to knock out genes or change the text, can alter the epigenome or the transcriptome of an organism of interest. And there's even other side-based applications for typing and tagging, antivirals, visualizations, targeted killing, and screening of natural events that enable those geneticists to do all kinds of things to manipulate DNA. But today, primarily, I will discuss about our ability to rewrite the genome for breeding, for ag and livestock, and engineering bacteria of interest to the food supply chain. So there's many reasons why this amazing and powerful technology has had the ability to democratize genome editing in less than a decade. However unfathomable as this may sound, is the many advantages that are exemplified and attributable to this particular technology, right? You can all see this. It's very programmable, very specific, transferable, efficient, precise, affordable, quick. You can do this within days, oftentimes, multiplexible and scalable, right? So our ability to rewrite the genome is really empowered. That being said, this is only an 8-year-old technology. So you can think of, the first cell phone 8 years in, or the first laptop computer 8 years in, or the first car, or the first boat, or the first submarine, or the first International Space Station 8 years in, right? It's not quite optimized as of yet. There's still many people working on sharpening, enhancing, and increasing the fidelity of those tools. And there's many ways to make it better and make it more efficient and really understanding and controlling DNA repair. And as with many technologies relevant for implementation in biological organisms, there's challenges inherent to their delivery and manipulation in different organisms of interest. But by and large, we are talking about maybe the most promising DNA altering technology we've seen ever. And though this technology is only 7 years old, our ability to use it and deploy it and harness it is unprecedented. And as a matter of fact, in the last 7.5 years, those 180,000 labs encompassing maybe about 1 million geneticists across the globe have put this technology to very hard work. We've been able in academia, for example, at the bottom of the pyramid here, to deploy this technology in simple organisms and model organisms. Think viruses and bacteria and yeast, or model organisms used by academics to understand and decipher organisms of interest as models. Critically for today, in the middle part of this pyramid, scientists in academia and also in industry have been able to show that they can readily and easily and affordably and conveniently and programmably alter the genomes of crops and vegetables and livestock of interest. You can see maize, you can see wheat, you can see rice, you can see some vegetables like tomatoes and the white button mushroom, but also organisms like chicken, right? And livestock, cattle, and even aquaculture, shrimp, and others. So across the tree of life for organisms of interest to food and ag, people have been able to deploy this technology and use it to rewrite the genomes of all those organisms of interest. And then, of course, as you can imagine, the medical industry, the biotechnology industry, the therapeutics industry has really made significant improvements given the many applications and implications for the use of those technology for medicine. And though this is somewhat out of scope for today's conversation, it's very important to know and understand and appreciate that the medicine and the medical industry, the medical applications of those technologies are serving food and ag, because it's enabling scientists to get access to better tools, sharper tools, more diversified tools, and that toolbox being enhanced and primarily designed for medical applications is being harnessed and deployed also by ag scientists and food and ag research scientists. So understanding the basics for the use of CRISPR-based science, turning this into genome editing technology and the many applications to rewrite those genomes of interest across the tree of life, we are already living in a world where some of those technologies have been commercialized, are being commercialized, or will be commercialized in very short order. And as a matter of fact, it's very important to know, as far as the bioeconomy in general and the bioindustry in particular, that multiple distinct and sometimes partially overlapping business fields have been fundamentally impacted by CRISPR research thus far. So there's a whole industry that is fueling the use and access of those tools and guides and enzymes and softwares and cells and kits that's enabling people to do this, which also means that researchers in food and ag don't have to make their own tools anymore, don't have to spend years to develop in-house insider knowledge about how to get access to those technologies, to use them in plants or livestock, but you can actually go to a website and order a CRISPR construct before 2 p.m. Eastern today and get it delivered tomorrow morning, COVID-19 distribution supply chain permitting, constructs overnight. It's that efficient, it's that quick, it's that accessible. And then those tools have been put to work in the biotech arena. So think of any bacterium, yeast, or algae used to ferment food, or biomanufacturing, or enzymes for household care and detergents, or the bioenergy and biofuel field. I will talk today about ag and illustrate how plants and crops and animals and livestock and bacteria are used for food fermentation, but also the forest, right? Trees, right? Flowers and aquaculture and beyond are being impacted. And again, as consumers, as investors, as patients, and as humans ourselves, we are going to benefit unquestionably from the many therapeutic applications that have been enabled and fueled and enhanced by the genome editing craze. And as a matter of fact, just 2 weeks ago was the first anniversary of the successful deployment of CRISPR-based gene therapies to someone named Victoria Gray, who received a single dose of a genetically engineered set of her own cells to address sickle cell disease. And as a matter of fact, there's over a dozen IND-enabled, FDA-condoned clinical trials that are recruiting and that are underway in the US and probably 24 clinical trials that are set to recruit people based on CRISPR-based technologies by the end of this calendar year. So it's very impressive. And again, this is important because those technologies will enable the food and ag industry and academics to use those sharpened tools for their own versions of genome editing. So here we are today. In 2020, we have already had multiple food and ag relevant types of organisms across the tree of life had their genome edited using CRISPR in the last couple of years to revolutionize how quickly, how efficiently, how affordably, and how flexibly we can rewrite genomes of interest, whether it's crops, whether it's fruits and vegetables, or even non-food crops like tobacco, or hemp, or cotton, or trees, or cellulose and bioenergy. There are a few examples here. The white button mushroom, Penn State, right? We can prevent and preclude the onset of browning by knocking out the gene responsible for white button mushroom browning. As a matter of fact, next year in the dairy industry, we will have the 10-year anniversary, not the 1-year anniversary like in therapeutics, but the 10-year anniversary of the initial launch in 2011 of the first dairy cultures that was screened for natural phage-resistant enhancement. And this has been commercialized globally across the world to manufacture yogurt at a very large scale from DuPont. Likewise, people have already proved a concept that you can target genes of interest involved in fat metabolism in pigs to produce leaner pigs. So think of maybe leaner bacon, for example, as a value proposition for customers. People have used those approaches obviously in crops of interest to knock out genes to increase yield, or knock in genes to increase drought resistance in maize. And then last but not least, you can imagine how genes that provide susceptibility of cattle in particular, or livestock in general, to certain diseases can be targeted to prevent the onset of disease. So in many ways, there are no limitations, perhaps other than our creative imagination with regards to what can be done with our ability to use, deploy, and put to work CRISPR-based genome editing technologies in species relevant for food and ag research across the world to enhance traits of interest to scientists and to industry. And as a matter of fact, this is very important to realize that this transition from a fundamental academic research-focused ability to do things in the DNA of species of interest has already fueled and is fueling a true paradigm shift for the whole food industry. As a matter of fact, beyond just the academic realm and the academic literature, we start seeing in the consumer literature examples of narratives relevant to food and ag that are illustrating and discussing and narrating very powerful messages to the readership about what can be done with CRISPR. So Alison Van Inneman, for example, out of UC Davis, a land-grant university institution on the West Coast, has illustrated and shared stories of how those technologies can be used to generate hornless cattle for more humane treatment and management of our livestock. Don't think of just the benefits for industry in terms of yield and financial benefits, but a more humane society, right? I mentioned in the last slide the work that I've done with others at DuPont to enhance the sustainability of the dairy industry by promoting resistance against bacteriophages, viruses of bacteria that are rampant in the dairy industry, to increase the yield, to increase the quality, and to increase the lifespan of the best starter cultures across the globe used for cheese and yogurt fermentation and manufacturing globally. And then some people like my friends at Lipman, of course, from the Harvard Lab on the East Coast, are illustrating how we can use CRISPR to target non-coding parts of the genome to really enhance the yield that we get for things like tomatoes. And you can imagine now a narrative talking about our ability to breed back into commercially relevant germplasm for tomatoes, the heirloom tomato, or bringing back flavor, or bringing back yield, or increasing the affordability of fine foods of interest, again, across the planet. So whether it's dairy, bacteria, whether it's crops, or whether it's livestock, we now have the ability to truly alter the genotypes of interest for functional gains of value, not just to the industry, but also to consumers and customers. And this tech push right now in 2020 and predictably in the next 5 years is going to revolutionize the bioeconomy. We are seeing in real time the CRISPR craze that all the academics like me, all the scientists like me, all the university professors like me are excited about and crazy about and enthusiastic about. We're seeing that basic science and technology come into the main consumer realm. Every week, right, one of those magazines discusses genome editing. Sometimes even features it on the cover, right? 2 years ago, as a matter of fact, on the July 4th issue, the most distributed issue of Time magazine featured genome editing on the front cover. We're seeing this, again, in NPR almost every week. I mentioned a couple of Wired stories, but globally as well with the BBC, and now even at a movie theater, we would hope near you, but COVID-19 decided otherwise, but you can actually stream on Netflix and online features by Hollywood, high-end, produced by professionals and Emmy Award laureates, narratives about how genome editing is changing our world as patients and as consumers. But besides the scientific craze and besides the media excitement and craze, what we're really witnessing right now is a new type of industry, is real investors realizing that there's potential for this disruptive technology as an investment. And the food and ag industry is a major player in that realm. So those are now not just the countries where CRISPR is a major player, but actually the investors putting in literally billions of dollars, primarily in the US, to establish companies that are hinging and founded on CRISPR-based technologies. So the very top here, the first generation CRISPR technology companies, Caribou, Intellia, CRISPR Therapeutics, and Editas, 3 of which are on the NASDAQ, having a good day today, NTLA, CRSP, and Edits. And they were quickly followed by second generation therapeutic companies like Beam, also on the NASDAQ. I think Casibia, Mammoth, Sherlock, very interestingly, has been able to show that CRISPR-based technologies can be also used to target RNA and actually detect COVID-19. Right? So we're seeing companies like Mammoth, like Sherlock, being able to work with big pharma in real time, within 90 days, show you can use CRISPR-based technologies to develop kits to detect a current pandemic we're suffering from. And there's all kinds of next generation companies, including one that was just announced last week, that are looking at the potential of rewriting the genome to enhance human health. There's a bunch of companies at the bottom here showing how you can use CRISPR as next generation antibiotics, as an alternative to broad spectrum antibiotics, to be programmable sequence specific antibiotics. At the bottom left, we're seeing a very fast rise in new companies that are focusing on developing the tools and the toys that other people will use. And then certainly last but not least, CRISPR as a breeding game changer for food and ag. And you can see right here how the big 4, right? The 4 large remaining big ag company, Corteva from Dow DuPont, Bayer, who took over Monsanto last year, BASF and ChemChina's purchase of Syngenta a couple of years ago, those 4 companies owning the large majority of the commercially relevant germplasm being planted across farms in the world, all have strategic plans for CRISPR and how they can use genome editing to breed next generation crops. This is also happening in the livestock world with companies like Genus And now we've also seen next generation new startup companies like Inari, Pairwise Plants, and Trico bringing those CRISPR-based technologies to compete with big ag because it's so accessible, so democratized, and so feasible to build value and create next generation companies to feed the world and fuel our bioeconomy from an ag standpoint. And as a matter of fact, it's not just me giving you my opinion on the field. There's a recently published report from the World Economic Forum entitled Innovation with a Purpose, the Role of Tech Innovation in Accelerating Food Systems Transformation, with a really focus on inclusivity of ag, sustainability of ag, efficiency of ag, and better nutrition and health for the whole globe. They feature CRISPR heavily and prominently and illustrate how this technology can be a game changer for breeding. Again, I'm not going to go into the details, but you can see that there's only two primarily fueling advances in science, one of which is next generation biotechnology and genomics, namely CRISPR, right? Creating effective production systems. Now we're talking about a powerful science and a powerful technology fueling effective food systems to really generate and rewrite the genome here in their own cartoon, right? Multi-trait seed improvements to generate up to $100 billion in additional farmer income across the globe, increasing productivity by 400 million tons, and changing the traits of some of those crops to address, health and nutrition, and in this particular case, micronutrient deficiency by up to 100 million. So we can see the promise of the technology from a scientific standpoint. We can see the promise of the technology from a technological standpoint, an investor standpoint. But what really stands between the true potential of CRISPR and genome editing and the benefits from society for consumers, for patients, and also for investors is acceptance of technology in an era, again, dare I say, where anti-science bias and scientific skepticism is arguably at an all-time high. So we now have a great technology that is extremely powerful and promising and deployable and implementable in industry and academia. But there's always concerns about how is this technology being used in humans? Is it ethical? Should we edit human germlines? How accessible is it to other people? Should we have a moratorium on certain types of research across the globe? Of course, besides the ethical issues and concerns for users and for society, there's PR issues. Is it a GMO? Is it not a GMO? What narratives are beneficial? Is it about a more humane livestock breeding and more manageable, or is it about industry profits only? So what are the key opinion leaders saying? And there's a tremendous opportunity at this very point in time to start strategically communicating to policymakers, to regulators, to investors, and to the public how those powerful technologies hold so much promise. And I'm going to commend Sonny Perdue and the USDA in aggressively and reasonably and having science-informed decisions and updates in policies with the secure rule that just came out a few weeks ago, enabling the recapitulation and the exploitation of CRISPR for a number of applications. So single nucleotide, single letter changes, deletions of any site, and again, the recapitulation of any genotype present in the natural gene pool of a species of interest without having to go through a lengthy, time-consuming, resource-draining natural breeding process to recapitulate the same outcomes, nonetheless, that would be indistinguishable from a genetic standpoint. And as a matter of fact, this is a very powerful tool in an ongoing effort across the ag industry to rebrand and update some of its perception, right? Going away from chemical-based product development and research and technologies to more biological-based technologies. Again, there's ways to use CRISPR I can go into if you're curious that do not necessitate the use of DNA. So think changing DNA without using DNA, harnessing natural repair pathways and non-transgenic material that do not carry any foreign DNA to change the genome of those organisms of interest. Again, it's also an opportunity to bring back our ability to exploit and use and recapitulate biodiversity throughout the globe in species of interest, right? We all know what corn is today isn't what it used to be tens of thousands of years ago, but we can bring some of those variants back into the gene pool using CRISPR-based genome editing. And then last but not least, we also now have the ability to really do a large-scale experiments and select at a larger scale variants of interest to breeders. And this means that at this very point in time, scientists and industry and business and political leaders like you have the ability to think about how to best frame this dialogue, right? What values are driving the development and deployment of those technologies? Again, in an era where people are skeptic about science in general and scientists in particular, we have a technology that provides new solution for more affordable food, for a more environmentally sustainable food supply chain. We can work with farmers to explain how this is going to be better to breed and manage our livestock in a more humane manner, or better manage the resources and the land and the water that we have available to us, again, to be more sustainable. And in the end, there will be tangible consumer benefits with their food systems, with their food supply, to be safer, to be healthier, to be more accessible, and to be more affordable. And this is where scientists, science leaders, and academic institutions, and the National Academy of Sciences, and thought leaders, and personable, likable, trustworthy individuals maybe have a responsibility to work on messaging, to promote environmental stewardship and fighting off disease and promoting nutrition and enhancing the organoleptic attributes of our food supply chain domestically and also globally. And at the same time, generate value, right, for the industry and investors involved, and promote consumer trust at the same time. A tall order, but a unique opportunity to do that right now. And as a matter of fact, if you, in closing here, if you step back just one level and look at what's happening in the CRISPR world, globally speaking, as those technologies get deployed to save lives in pharmaceuticals, we can piggyback on therapeutic success to advance acceptance of that technology. As the public becomes enthusiastic about CRISPR to fight disease, they are more keen. Maybe it's more palatable to them to use the same technology to enhance food. Of course, this has to unfold in an international geopolitical context where Europe, especially with Big Ag, with BASF and Bayer being a major player today, but as well as China rebalancing the US and the Midwest, historically perceived as the leaders in Big Ag, they will catch up. They are catching up, they're trying to compete. And the industry, by extension, is going to invest in those spaces. So how competitive are we right now, and how competitive do we want to remain and move in foreseeable future? As the ag world consolidates and diversifies, how are we going to deal with this? And of course, it's going to trigger actions at the USPTO level with IP drama with CRISPR. It's going to trigger investors to promote deals and investments and mergers and acquisitions. And in many ways, the ag industry has been revolutionized just with M&A in the last few years. And then at the same time, there's opportunities for regulators like the USDA, as we saw in Australia with the secure rule, to promote the use and exploitation of science as best as possible. And of course, I think because it's science-driven and is very research-intensive, ensuring how we manage control and dissemination of intellectual property in an international geopolitical context is ever important, sensitive, and critical. So I'm hoping that this is a good introductory overview of where the CRISPR craze is today, how academics are advancing the science and advancing the technology being applied by industry, being fueled by entrepreneurs and investors, and being used by farmers to generate products disseminated and endorsed by government and politicians and regulators and the patent office to really eventually benefit society the public in general, but consumers, patients to some extent, and then work on messaging and PR to ensure that the future of food and ag is accounted for and enabled as best as possible. So I'm going to thank you all for your time today. Quick apologies for the missing out the first slide early on, but I think you got the gist of the messaging. Thank you for your invitation. Thank you for your interest. And I think we have plenty of time to field questions at this point in time. So I'll bring it back to Clint, and we'll field questions for the next 20 minutes. Thank you very much.
Very good. Thank you, Dr. Barrangou. So yes, at this point in our program, we would ask that you turn to the question function within the webinar. Please type your questions there, and I will be able to present them to Dr. Barrangou in the remainder of our time. We have a couple of questions already in the queue, so I will read those to you. The first question is a question about CRISPR products and other forms of biotechnology, including transgenics. Do you believe that that biotech may pose a limitation to consumer acceptance to CRISPR? Now you touched on that a little bit in your presentation, but maybe go back to that point again.
So yes, so I think we know, and the scientific data has shown that the public is averse to science in their food in general, and is very concerned, or a population of the public is very concerned with genetically modified organisms in particular, especially transgenics, right? Taking DNA from another species and put it into a species that we put into our mouths and into our body. So this is certainly a hurdle that the ag industry has had to deal with since the 80s. This is a narrative that we haven't been very good at selling and at pitching and at articulating and addressing. But in many ways, CRISPR can provide a unique opportunity and solution to that problem, because technologically and scientifically, there's many ways to use CRISPR without the use of DNA to recapitulate natural events that would occur as part of breeding that are indistinguishable from natural breeding processes. So we're talking about non-transgenic, non-GMO, non-genetically manipulated organisms that are natural in many ways, or equivalent to those natural variants. And then this is where, again, the US in particular, with the USDA secure rule, has laid out a very enabling and promising, encouraging framework that other people are paying very close attention to promote the use of that powerful technology to generate products hopefully acceptable to the public. So yes, very important, still concerning, but a clear path forward in terms of PR.
So then I think a related question to that would be that to some extent, a lot of the talk about CRISPR and the use of gene editing sounds similar to the excitement around the older transgenic technologies from 20 or 30 years ago. So what do you think that scientists and communicators, public officials and the like can do this time around that's different than the way things went 20 or 30 years ago? And
yeah, so the world is changing so fast. I mean, this is very easy argument to make today, right? The pace of change, the rate of change, the impact of change in the world in general is maybe the biggest it's ever been before. It doesn't make people comfortable. It comes with caveats and challenges, but I think people understand that the world is changing fast. And technology likewise is changing the world and creating new solutions to the problems that we have. So if you think of the world 30 years ago, the world was very different. Technologies were very different. And CRISPR is a great example of how 30 years have changed technology. So equating CRISPR today and genome editing to transgenic technologies in the 80s is inappropriate and scientifically dumbfounding and unsound and incorrect technologically. So we now have new technologies that enable us to do new things in new ways that really don't compare or contrast to what we used to do at the time. And with that powerful and flexible and nimble technology come options to do things that we could never do before at a speed, at a pace, and generating outcomes that are naturally recapitulated. So we're talking about changing things to mimic what nature is doing, right? Now, of course, there's ways to use CRISPR that are transgenic. There's ways to use CRISPR that are truly GM, which is why it's hard to really comprehend what the differences are. But at this point in time, the ag industry is really focusing on manipulations, applications, exploitations that fall within the natural realm.
Thank you. Another question from the audience. How has the recent pandemic and COVID-19 changed, if any, potential uses of CRISPR?
Big time. I mean, this has been inspiring for team science in general and team CRISPR in particular, in real time to redeploy our most capable solutions, technologies, people to provide solutions to today's problems. So I mentioned Mammoth, I mentioned Sherlock. There's multiple companies that have actually worked on detection kits for CRISPR within 90 days. I mean, 90 days ago, it would have been unfathomable or a dream come true, literally, ever since. There's people using CRISPR technologies to work on vaccine, right? There's almost 200 different vaccine projects across the world. There's many of those that are hinging on CRISPR-based technologies to develop antivirals. As a matter of fact, CRISPR can target RNA and target SARS-CoV-2. It is like, it hasn't been shown in humans yet, but there's enough proof of concept provided for a Cell paper last month to say it's a worthy avenue of research. So we're seeing this. Now, besides the science, what this is doing is it's increasing the appetite of investors for food and ag, right? All the money you put in transportation and vacation and travel and tourism has to go somewhere else. And, people need to eat every day. And we now have the ability to fuel next generation food and ag, to invest in it, to not just deploy this technology for genome editing and therapeutics, but to develop this technology for genome editing in crops and in livestock and in fermentation bacteria and in next generation enzymes. All the things that are biological that touch the food supply chain can be beneficially impacted by CRISPR.
Thank you. So speaking of livestock, you spoke about USDA regulations on plants that could have been produced through conventional plant breeding, but this is very different than the way FDA regulates gene-edited animals, where they consider all intentional alterations as being regulated as animal drugs. Could you comment on how this might impact democratization of the genome editing that you discussed earlier?
Yeah, I think it's a question of pace, right? So there are different timelines and regulatory processes that apply to different types of organisms. And as you mentioned, and as I said earlier today, I think the secure rule rolled out by, again, Secretary Perdue and his team at the USDA is very encouraging and very science informed. And I think it will take a little bit more time for people to attribute the same benefits and come to the same conclusion with livestock. So to me, it's a question of when rather than if, and a question of how fast versus how slow. Now, this is what I call the highway of science, right? So there's people at the cutting edge of the highway that are blazing new trails so we can build new roads, right? And those people with CRISPR are going so far, so fast, off-roading occasionally, right? But they're blazing those new trails. And as they create those new avenues of research, as they create those new roads that we can embark on, then the asphalt comes down, and then eventually people come in to say, that's the speed limit you should use. You have to wear a belt. You have to have a speed bump. You have to have guardrails. So the point here is that regulators can only come in after we are well engaged in how this technology can be deployed, and that kind of discomfort that we have between where we are today in academia and where we're going to be tomorrow in the public, sometimes it takes years. And we have to get comfortable and trust that the agencies in charge of policing and regulating and endorsing and assessing those technologies are trustworthy in doing their job, and eventually will enable industry to have the right applications that are safe and reasonable for consumers to eat and consume.
So you feel that in a sense that the regulatory system will sort of lag behind the development of new innovation to to catch up.
Yes, but by default, right? Science has to happen first, right?
Right.
So can you actually do it? Because trying to regulate it before we can even do it is a futile exercise. And this is what I love about CRISPR, because it's making people uncomfortable, and it's good because we can now do so many things, and it's becoming such a reality that we have to have a conversation for real, as opposed to what could we do with it. Is now that we can do this, how are we going to regulate that? So it's making people more uncomfortable. It's making some people anxious. It's making some people perplexed. It's raising issues that we're very familiar with, but it's making them very real. And we need to have a sense of urgency to address in a timely manner the balance between safety and confidence for the public and consumers and aggressive pursuit in a competitive world geopolitically to be at the forefront and to get financial benefits as a society, as an industry, as a nation that comes with it.
I think while we're on the topic of livestock, there's another related question. Is there anything scientifically different about the risk associated with genome editing in food plants that's different from genome editing in food animals?
I mean, overall, if you look at it from a global standpoint, not really. There are small details about the molecular machines that are being used. So, for example, some of those I don't want to get too technical here, but some of the SP-Cas9 that comes from a human pathogen can trigger more immune responses in a mammalian or animal type system than it could in a plant. So in terms of toxicity and efficiency and DNA repair, there's some technicalities that are different. There's some reactions that could be different, some adverse effect that could be slightly different. But by and large, if you don't really look at the scientific details and you look at the overall ability to deploy that technology to do breeding by editing, the same principles apply. And this is why advances in therapeutics are so critical for livestock, because what we know works well in humans is that much more likely to be portable to livestock. Plants are much easier. I don't want to trivialize that, but plants are relatively more straightforward to work with than livestock.
So another question from the audience. In the diagram of who consumers trust for information on gene editing, I don't see any mention of social media, friends, consumer advocacy organizations, etc. These may be sources of information that some consumers trust, and that may suggest that gene editing is unsafe, driven by profits, or otherwise undesirable. Are these alternative trusted sources of information a consideration? How can or should these sources be addressed by researchers, companies, and the like?
Absolutely. So I don't consider necessarily those sources of information trustworthy. And this is the challenge. There's a lot of misinformation or disinformation. I'm not going to get to that tangent, preferably in the next 7 minutes, try to stay out of trouble here. But I think there's rightful scientific skepticism about unwarranted media and social media coverage of science. So it absolutely has to be accounted for. It absolutely has to be managed. The scientific enterprise and the industry need to account for this in the development and deployment of strategies for communication, unquestionably and absolutely. But I do not consider them systematically to be trustworthy, and that's the challenge that we have. So to me, the solution will come from trustworthy sources using the right means to disseminate the message, right? So I'm not going to do a TikTok for CRISPR and genome editing today, but some people should. I'm not going to do, my lab is an Instagram account, but somebody's running it for me in my lab. Like I would spend my time doing this, but some people want to see that. You need to have your Facebook page, and you need to have your LinkedIn profile, you need to have your Twitter accounts and whatnot. And we absolutely do have to do that. It's part of the picture, but it requires its own strategic impetus, but it's not necessarily by default a trustworthy source of information.
So as you know, the main function of NCFAR as an organization is to really support and advocate for national funding for food and ag research, extension, education, and the like. Could you give us some idea about not only how public investment may have supported your research or research in this area, but also more generally how you see the balance between investment from private sources relative to public sources?
I mean, I think I'm a great example of this, right? I'm an employee of the great state of North Carolina. So I'm a state employee at a land-grant university, state institution. I'm an educator. I teach. So I'm being paid by the taxpayers' money to teach and instill knowledge and expertise upon the next generation of ag specialists. And I also perform research. And some of that research funding comes from the USDA, some of that research funding comes from NIH, some comes from the NSF, always not enough from federal sources, and a significant portion of the budget in my lab comes from industry. Because the solutions that we have are immediately implementable and exploitable in the industrial realm. I spent 10 years in industry. In my past, I also went to business school, so I understand the impetus and the diversification of the portfolio value from a funding standpoint. So I think it's got to be a team effort to answer your question, some of which has to be state-driven, state institutions, land-grant universities, and more elect, some of which have to be federal, absolutely. And as a matter of fact, this audience knows very well that maybe unquestionably some of the therapeutic funding should go more to ag because it can make a huge difference and have a real impact. So I'm not going to be that horse because the audience knows this already. But also finding ways to be more collaborative with industry is critical. And I do that certainly every day. A lot of my fellow CRISPR scientists do that every day. And to me, it's important not just to advance the science, but to stay competitive domestically. In a world where geopolitically we're competing with China for IP, for technology, for investments, for food supply chain, and in a world where our strategic partners of today may become our competitors for tomorrow, right? I mean, Europe is a big player in genome editing in food and ag. We know how their consumers and their regulators feel about GM and CRISPR and editing, but this will change. And I think we need to fuel now more than ever. We're at a very critical point in time where if we want to stay ahead of the curve and remain number one and be more competitive geopolitically, we need to account for it and plan for it and strategize accordingly. And industry and government and academia need to work in concert together to fuel this and make sure that we maintain our ability to stay at the forefront and preferably stay in the leading position for genome editing in food and ag and beyond.
Thank you. I have one last question in the list, which is actually fairly related and nice segue to the comments that you just made. This question is with regards to whether or not the technology is being addressed in trade negotiations and I assume international regulations as well. How does that affect the picture?
Yeah, so the WHO is working on this. The UN is looking at CRISPR. There's a lot of international organizations that have their ears to the street. International National Academies are a member of the National Academy of Sciences, National Academy of Engineering, National Academy of Inventors. So all those national academies are publishing opinions and working on the technological landscape and ethical use, potential risks and abuse. So absolutely, we're all informing our respective governments with regards to what they should do and how they should do it. And again, I'm going to commend the USDA and our commissioner, Sonny Perdue, for being aware of it and having a milestone policy update, 33 years overdue earlier this year. But we have to be very mindful of the fact that there's other people having that conversation right now in other time zones that are trying to be competitive. So can China buying Syngenta is an example. 30 by 2030 for Russia, right? They're investing 1.6 billion euros to generate 30 species being edited by 2030 in Russia. I don't know how they're going to do it. That's a different conversation, but we absolutely have to be ahead of the curve geopolitically. And there's huge things at stake and in play. And managing that as part of the world trade with the global food supply chain that is really shifting when it comes to livestock and crops. We know how much import and export is in play. It's absolutely part of the international trade organization conversation.
This is certainly an area that bio plays in as well. And certainly our experience is that many countries around the world now are working through what their regulatory systems are going to look like for these new technologies. Does it make sense to use the old GMO things that have been stuck that way for 20 or 30 years, or think outside the box? And thankfully, we are starting to see a lot of countries that are leaning more towards a USDA-like approach where it is more efficient, more streamlined, and more science-based for some of these newer technologies. But I think you're absolutely right, and this is sort of what we're watching as well, that how that map of regulatory systems around the world plays out will significantly affect trade, and where the U.S. ends up in terms of competitiveness in agriculture around the world, for sure.
Absolutely. Could not agree more.
So that was the last question I have on my list. We have about 1 more minute, so if anybody else wants to squeeze in one additional question for Dr. Barrangou, I'm happy to take that.
And I'm very easy to find. So send me a note, follow up. Thank you again for your time and the invitation. I look forward to maybe a future opportunity to do this in person. Thank you.
Thank you, Dr. Barrangou. I'm now going to give you just a little bit more information about our upcoming webinars. If I'm able to switch to the slide, give me just 1 moment. For some reason, I am not. Otherwise, I will just give you this information verbally. I apologize with the technical glitch here at the last minute. But I just want to let you know that there is another NCFAR webinar that will be scheduled in September. We will be focusing on, excuse me, just let me get my list. We'll have a webinar about the new National Bio and Agro Defense Facility, which is going to be the nation's premier animal disease research facility. So watch for your invitations. Stay tuned, and we ask you all to have a nice, safe, healthy rest of your summer, and to please pay attention to additional invitations from us. So again, thank you, Dr. Barrangou, for joining us this afternoon. We appreciate everyone's participation, and this concludes our program for the day. Thank you all for joining us.